First of all, thank you so much for having me. I am wonderful to be here, albeit Zoom. Um, I was telling Sarah previously that it was very, I don't know, interesting for me to write this talk because I kind of summed up a lot of what I have learned over my career. It's not coming to an end, just the mid career. And suddenly I woke up and I noticed it was like over 100 slides. So I asked Sarah if I could cut it in two. So, in order for me to speak slowly and to allow questions, whenever we stop at the end of the hour, we'll stop and I'll pick it up. I'll coordinate with Sarah. Part two, shall we speak? Please feel free to ask me questions in the middle if something's unclear. And the second thing I wanted to mention before we start is the reason I really wanted to bring this topic because all of you do a lot of more recent stuff in the general sense. The Americas heritage is much, much more recent. And in the Levant and in Europe, etc., we have things going back millions of years. So I'm working on a site right now that's 1.6 million, for example. But I think that there's a lot to learn from the methods that we use in deeper time with modern heritage or analysis in archaeological sites that are Holocene in nature or even mid-Holocene. And so I wanted to bring this as kind of a discussion point to think about methods that we could apply in the United States for different projects and stuff like that. So my title is Climate and Environmental Change Across Temporal Scales, an Insight from the Southern Levant. See, here we go. 1 minute. There we go. So basically what we're going to do is I want to kind of introduce you into Pleistocene ecology. And I'm going to talk about a very interesting phenomenon that most people are not aware of called the Pleistocene paradox. And the Pleistocene paradox basically, and I'll show you in a minute, kind of is an unknown why the patterns that we see in the early Pleistocene and earlier time periods, pre-Pleistocene, don't look at all like the climate change patterns of animals, etc., that we see in mid-Pleistocene and Holocene and late Pleistocene. We're going to focus on mammals, or specifically mammals, but in general animals and not plants. Obviously, there's a lot of different types of organisms that respond differently, but our focus here is going to be how animals respond to climate change, and we'll look specifically at three methods. The skeletal changes, something called dental ecometrics, which encompasses mesowear and microwear. And finally, I will present my work in the Southern Levant and kind of use it to integrate everything together. So the Pleistocene paradox was coined by Sterenli in 2001. And it's basically defined, as I mentioned, as it's a discrepancy in community response to climatic shift between the Middle Pleistocene and Holocene, where the pattern is what we know as Glissonian, and the Pre-Pleistocene Clemensian pattern. And when we look at more analyses, we can see that the ecological mechanism that holds communities together differs between more paleontological and neo-ecological assemblages. Sorry, decided to. So just kind of focus on when we think about the general idea of a community, the main questions that still exist are whether it is a kind of abstraction made by ecologists, do we have a real biological ecotones that are regions of rapid replacement in the more Clemensian method, or do we have these open communities where species kind of change independently and we don't have a cohesive community that's kind of moves along evolutionary. So one thing to remember is that we have a phenomena in the Pleistocene or even Miocene and Pleistocene of coordinated stasis. The coordinated stasis is basically a bimodal distribution of community level stasis that can be, for many, many millions of years, and then suddenly there's a rapid turnover. And when I say turnover, I want to clarify that I'm talking about also faunal turnover, like taxonomy. If we have a mammoth, one species of mammoth or one species of bear, and then suddenly there's another species of bear, that's a change in species membership. We can also look at ecological structures like alpha diversity, which is like number of species, relative abundance of different species, guild representation. And we have to kind of mirror this with Stephen Gould's hypothesis of punctuated equilibria that talked about really a similar pattern that we see in long-term morphological evolution. So how to explain this Pleistocene paradox, right? So one theory that has been put forth is that the ecological mechanisms that hold communities together differ between Pleistocene or paleontological and neoecological assemblages, and that there's really something there that we need to explain about the response to climate change. And the second point is that either, coordinates say this is just a taphonomic artifact of time and, space averaging. It's entirely a function of our resolution of how we're capable of studying um, uh, faunal assemblages in the past. And third, we all know that the Pleistocene is known for the change in amplitude and frequency between inglacial and interglacial periods. And perhaps just the amplitude and frequency between these pre and post Pleistocene communities is enough to explain this difference in patterns. And I will jump to the end and tell you that I don't have a solution at the end, but it's an interesting point to discuss. So this is just a nice representation to show you the timeline. And we have these deep time paleofaunas and paleofloras that are in the millions of years. Most of our faunal assemblages are actually in the kind of late Pleistocene record. And then we have modern extant communities, obviously. And again, we can talk about the succession of communities, species invasion, extinctions, and speciation over this time period. The difference is also how do these faunal assemblages accumulate, where more recent studies kind of focus sometimes on mass death or event assemblages, and we have a lot of time and space averaging in the more deep time. So this is just a pattern to show you. This is one of the first papers from Brett et al. In 96. And you can see here, this is the Middle Devonian. I know nobody here is working on the Middle Devonian, but I thought it would be a nice show that it works there. And we can see this community that is typified with these kind of brachiopods and various invertebrates. And we see suddenly in the Gavetian, that there's a complete change in the species distribution to change again towards the end of the sequence. And that is very, very different from what we see in Pleistocene communities. So this is the last glacial maximum, where we see a Glissonian community structure. And here's just for an example, they did a lot of work on pollen and trees. And we see that through the last 20,000 years, the distribution of different species occurred kind of individually without one species, without a community moving as one together across space. So the questions that we'll kind of talk about, at least in this part, is the Pleistocene paradox, and basically, how do we measure climate change, or how do we measure this response? How can we, as archaeologists, look at our record and understand this thing? The second is, how do we appreciate taphonomic patterns, right? To try and find the parallels between evolution and ecology, right? So individual evolution and morphology, and ecology as a community-based structure. And two very interesting hypotheses, the Red Queen and Court Jester hypotheses, that talk about what is the most important factor in actual community change. And we'll talk about resilience and response to environmental stress. So I don't expect you to read this this table from Berensmeyer in 2000. But what's really interesting is that when we look at taphonomy, there's a lot of potential biases that you can quantify in the fossil record, including, for example, maybe shifting in original composition, right, because of diagenesis and the biochemical fidelity. You can sometimes soft tissues, of course, are destroyed or bones are fragmented, and we have this physical deformation. We have spatial fidelity because of transport out of the original environment into a new environment. Temporal resolution when we have this mixing of several non-contemporaneous remains in a single unit. Again, we have episodes of deposition, and then we have diagenesis and obliteration, and we don't really have a complete time series to work with. And again, the consistency and preservation over geological time, what was good in the Cretaceous maybe not be good in the Holocene. So just to demonstrate this graphically, we can look at taphonomy here, and these are the ancient communities here represented kind of in a Clementsian pattern, which basically means distinct communities. And when they die, because everybody dies, then we have subfossil assemblages. But then what happens in the actual fossil assemblages is that we have both time averaging and we have a spatial averaging of these four, right? So we have a fossil sample that is really, really maybe not representative of the resolution that we can find in more neo-ecological research. For example, here's a paleoclimatic proxy, let's say oxygen isotopes. And if we look at a third eighth of a cycle, we can see that the averaging kind of mirrors this change. And this is kind of maybe the resolution we can achieve when we look at more neo-ecological assemblages. Or if you average it at a larger scale, we really see stasis. We don't see anything. This is the idea. It was republished by, this is a republication of Eldridge and Gould in 72 of the punctuated equilibrium model. And this is, of course, what they don't think. But a lot of early evolutionists supported the gradual evolution. And basically, you have a population that has a mean and a standard deviation around a certain morphological character. And as time progresses, this mean shifts, usually keeping the standard deviation the same. That's another issue. But overall, we can get to, as time passes, to a different completely morphology that's not affected in that we don't see basically in the earlier species. So, but what Gould and Eldridge suggested in 1972 is this punctuated equilibria, where you have a parent species which continues to exist in a certain morphology, but there's a very rapid evolutionary occurrence that leads to a daughter species somewhere else. And that's reflected in the fossil record by one morphotype that then suddenly appears another morphotype as if out of nothing, as if you know and that really contrasts this idea that you have this gradual change, species being a intermediate or a, and then we have the two other species. And the reason I brought this up is to kind of think how it mirrors the ecology. When we think about this comparison between macroevolutionary and phylogenetics and microevolutionary evolutionary dynamics selection, so we have these two, they actually kind of involve or interact with ecological processes to create what we call community function, which is basically what the structure is, how do they recycle nutrients, productivity, but primarily indices of diversity and richness and persistence over time. So this interaction between phylogenetics, macroevolution, what species it is, numbers of generation, population size, what you're studying, right? Clades, divergence time, and you combine that with this cyclical, different time scale of ecological processes where you can talk about species richness and composition and habitat type and spatial scale. And these two of course, are balance each other out in the long run. So the Red Queen hypothesis is a very famous hypothesis that was said by Van Valen in 73. And of course, it's named after the Red Queen in Alice Through the Looking Glass. And there, the Red Queen says that you have to just run as fast as you can, just to keep on running faster and faster, just to stay in the same place. And this was suggested that if you have two species that one is a predator, one is a prey, or they're co-evolving, each have to evolve in response to the other evolving just to be able to maintain that relationship. A lion's never going to evolve to eat all the gazelles. Um, and the underlying assumption from this for ecological change was that biotic interactions between species, competition, coevolution, etc., um, are more important than abiotic interactions in driving evolutionary force. But we come from a perspective of more climate change, right? And habitat change. And how does that change biotic interactions? And I think this is an important counter argument. So Baranski in 2001 developed the court jester hypotheses that talked about that abiotic and environmental change, which is what we're studying as a whole, are the main drivers of evolutionary changes in organism and ecological changes in ecosystems. And again, the court jester, the idea here is that you can have a perfectly stable homeostatic environment. The species love each other, everything is fine, we're all happy, and then suddenly a whack from nowhere, and you have something as, I don't know, quick as a tornado, an extreme tornado, or a volcanic explosion, or also it's extreme cold or Heinrich event, and then everything changes. So here, just to compare the two ideas, in the red queen hypothesis, the abiotic environment is kind of like the same, but despite the abiotic environment being the same, we see that the adaptive zone of these two species changes rapidly in this transition time based primarily on some kind of biotic interaction that changed along this place. For example, here we have the blue species is more abundant than the green species, and here it's kind of the reverse, and they begin to undulate a little more. In the court jester, we have this abiotic crazy sequence, but sometimes the changes are not within the realm of adaptation. There's a really very deep change, and we see that results in the adaptive zone changing completely from, let's say, the green species is more resilient, but the blue species become extinct, and the orange species kind of appears. To kind of focus on more very classical hypotheses, is Verba in 1972 developed her turnover pulse hypotheses, which again argued that climate was the most important instigator that affected major lineage extinctions during the Pleistocene. And then basically you had a turnover that happened in pulses that could be related to different changes in climate across clades. So this leads me to kind of a question that maybe I should have brought up a little earlier, but what do we mean when we say climate change, right? So here's a nice little diagram taken from physics to scare everybody, but basically just tells us what we're talking about. So between the crest and the trough and the next crest, so between crests is the wavelength, and the amplitude is between the reference line and whatever the crest is, or between the trough and the reference line. So we can look at change over time as that there's inherent variability, right? This winter is going to be probably warmer, then it becomes colder a little bit, then it becomes a little warmer. We can look at directional change when we talk about climate change in a directional way, that we see a consistent warming. We don't see suddenly, we don't see it as within the general gradient or range of change that we see. We can look at a step change. So again, you have biotic environments that change, but then you have suddenly a shift. Or more important for our argument here as extremes are shift in variability, right? The variability is very, very important. We kept on talking before on the same standard deviation, a change in the mean, but extreme shift in variability is perhaps one of the most, I would say now it's more looked at, but basically, I think not addressed as clearly as it should be. So going back to the temporal scales in years and spatial scales, we can look at macroevolutionary. So again, evolution above the species level. We look at continental formation, the impact of asteroids, right? This is a very, very long term. We can talk about of the KT boundary and the extinction of dinosaurs writ large, except for the birds. And we can talk about, let's say, sea level changes and climatic oscillations. And this is in the millions and more than millions of years. In the ecological or microevolutionary changes, and which is what you incur over usually your lifetime, but or maybe your, several generations. We can look at extreme wildfires, typhoons, hurricanes, floods, insect outbreaks, droughts, and heat waves, right? And each of these affect both the individual, but also ecosystems, ecoregions, and continents. And I think that's important to kind of reconcile both. Um, I think it's also important that we put into this equation these two mechanisms of adaptations. So one mechanism is an evolutionary adaptation. If everybody thinks of their classical evolution of the horse, and we see it goes from a three or a five-toed, small little browser, and it grows in size in a linear fashion as a response to the development of grasslands with a lot of phytoliths and quartz and other hard and abrasive elements. And they have their teeth grow much, much taller, and they lose their metapodials. They are on one toe, and they become much larger. Now, of course, today we know that that's not the exact pattern, and we know that horses, in their lineages, when we look at the large clades, some clades became smaller, some clades, did not lose all their toes, some clades, became a little more browsy. But if you think of the evolutionary scale, we think of processes of selection and mutation, and then you have the sorting based on the environmental change over multiple generations. But you also have the response of ecology. And what I tell my students and people is that if tomorrow it's going to be 80 degrees, which I won't be surprised if it happens, it's not going to be that all our cold-loving species are going to keel over and die and disappear within 24 hours. There is always a shift, first in behavior. It could be physiological. It could be that they start shivering, or they find shelter, or they do some kind of behavioral change that is not like, oh my God, climate has changed, I'm gone. Obviously, if there's more climate change, we begin to see this big, big change in, of course, multiple generations. But within generations, you usually look at phenotypic plasticity and migration. You don't like where you're living? You move to a warmer place or a colder place. I will focus a lot on the phenotypic plasticity that we see in this within generation changes in animals. And of course, in multiple generations, you do see species sorting, as we talked about in a Glissonian manner, and communities changing, and this is parallel to genotype sorting. So, what do I do? That was a big introduction, and I'll start talking about fauna and what we have in a minute. But we have to remember that, of course, paleoclimate, deep time paleoclimate, like stable isotopes or deep sea cores or ice cores or soil carbonates or speleothems, of course, intersect with our knowledge of fauna and knowledge of flora from the biotic environment. And as I said, we talk about in the community structure, not in the own morphology, between the presence absence of species, something called rank abundance. So, what species has the most abundance is going to be ranked as one, two, three, four. And sometimes, even if the actual abundance is not significant or changes in a way that we can appreciate, the rank abundance may stay the same. So, they shift differently. And we have ecomorphology, which is differs from ecometrics, and it's kind of confusing. But ecomorphology is changes in the general morphology of animals, and to see their general adaptations of the whole species to an environmental ecological scenario. So, as we spoke about horses, equids that live in plains with grasslands. Ecometrics is more changes again within I'd say individuals or populations, so it's below the species level, of changes of metrics of actual measurements in the teeth and body that reflect what habitat did this individual actually live in. So obviously, you can be a big horse with great teeth, and if I put you in the tropics, you won't be eating grasses, right? You may eat some browse. Now, it's not optimal for you, but you'll survive, depending, of course, on the deviations. But in general, ecometrics says, look, we have a fossil. What did this fossil eat actually in the environment? Not it's an equid, there are grasses in the environment, but let's look at the teeth of the equid and try and understand what was really around us. And kind of to continue in this line, we also have different degrees of response of animals. So there could be some climate change, and the animals do absolutely nothing. They don't change. They're adapted to this range of variation that exists in environments that they can still produce. We also have these behavioral changes that I talked about, which are intraspecies ecometric diet, like diet. We can also have behavioral changes that are more at the population level, like demographics and range change. The third level of response is within species because they moved to another place or died. You have a change in taxonomic turnover, but no change in what we call the functional diversity of an ecosystem. So, for example, if it's a temperate woodland in North America, the deer species, specific deer species, may change, or again, other species, but the actual idea that it's a temperate forest may not change. And finally, the greatest degree of response is a taxonomic turnover coupled with a change in ecosystem or functional diversity of the community. Another thing we have to remember is that different animals, based on their trophic level, respond differently to these differences in amplitude and degree of climate change. So while, for example, in phase 3, carnivores tend to be the most static or more the most persistent in their behavior compared to herbivores and primary producers. So, for example, plants would move, of course, will be respond much more abruptly to climate change than deer because deer or equids or herbivores can consume different types of vegetation to some extent without it causing them grief. Carnivores just eat meat, and I don't know if they really care what the taxonomy that we imposed on these deer is going to affect their diet. There are some exceptions to that, and we can talk about that later. Again, size. And this is where I like to say that the difference between small mammals that are very rapid to response because their generation size is very short or length is very short. They have a small a home range compared to large animals that are usually very persistent. Think elephants, for example. So here's what we commonly think of major climate changes in the Pleistocene. And this is just a reconstruction from uh, in the last glacial maximum from on the Bowling-Gala Road and whatever. But we go from a very, very tundra-like with certain species to a warm and swampy period. And so you have both a taxonomic shift, right? The first, the proboscideans on the top picture are not the proboscideans here. And we have an increase of new species and no rhinos, for example. But we also change the taxonomic diversity together with functional diversity. But we have changes, as I mentioned kind of briefly before, changes in taxonomy that can maintain functional diversity as well. Let me see. Okay. Yes, we're in the right place. So Peter Andrews was one of the pioneers in looking at communities at different taxonomic diversity, but are similar in functional diversity. And the idea here is that if you go to a Pleistocene or Miocene even faunal assemblage, you don't taxonomically identify the species. I mean, you could, but I'm talking about you don't know where they lived because it's a clade that's unknown to you. And you can't use the common method of combining or comparing the closest living relative to the species. So a lot of our understanding of equid ecology is basically saying a fossil equid ecology is saying modern horses look like this. This is their tooth structure. When we find a fossil that has a similar tooth structure, we infer not only that it's an equid, but also that it eats a lot of grass. And what you see here are four different, very distinct habitats in Africa. But you see here that he divided into four main ecomorphological categories. So this is size, A, B, C, and this is the abundance of the number of species. So if you have, um, let's say, 50 species, 40 are going to be of the small size, and so on and so forth. And you can see in these silhouettes that there are differences between a flood forest, for example, and maybe a short plain Serengeti, right? And between a lowland forest. And you can see this very well that we have a Cebosipella and Trachypithecus so one is a New World monkey, one is an Old World monkey, and they occupy the same functional position in the ecology, right? Despite the fact that they're very, very distant taxonomically. Here's another example of something that happens. This is from a site in Atapuerca near Barcelona, dated to about 1 million years ago to 500,000 years ago. And we have the lower Pleistocene and middle Pleistocene. And this is our ecological change or the distance between, let's say, abundance of species, right? And we see a relatively flat. There's no change between the lower Pleistocene or isotope stages 27 and earlier. Here we have the Bruns-Matuyama reversal and the reversal in our magnetism. But we do see here in this level equivalent to MIS, let's say 16, between 16 and 17, a complete change in taxonomic turnover, a complete change. But the ecological change of number of species and each, how many species are carnivorous, how many species are herbivorous, what is the size distribution of the species, doesn't change at all. And that is kind of the core of what I'm trying to get at, is that change is not an all or nothing issue, right? And we have to start parsing out change. And this is where I would like recommend to kind of think about where our more modern communities are going, because we can expect this discrepancy, right, between how you identify a faunal collection or what you see and how you're interpreting the change. So, for example, this change, I don't know if it's related to any major MSM changes, MIS isotope changes, right? Because it happens at a certain point of time, but then you have these long periods of stasis again. So stability in the guild structure. And we talk about behavioral changes. So before, you're stable for a while, but then things get really, really rough. And you can change what you eat. And we have four scales in which we measure these dietary changes. The first one is the time of the enamel deposition of your tooth. It can be right after birth, before birth, and we usually measure these in stable isotopes, specifically carbon, in later time periods in nitrogen, either as oxygen. A week before an animal dies, the microwear that's in its teeth. So every time you eat, the mechanical properties of the food that you're consuming scratch your teeth and create these micro patterns of microwear. And obviously, if you eat hay one day and then eat browse one day, the browse will overwrite the graze or the graze will overwrite the browse depending on what direction you're going. But 1 week before your death, it's usually not erased, and you can figure out what a specific animal, not even population, a specific animal ate right before it died. 6 months before death, you have something called mesowear, which again reflects the dietary adaptations of animals over, let's say, a season. We're talking about, let's say, larger species, so they have several generations. Obviously, 6 months is not a life cycle. And then finally, we have in the evolutionary scale where we look at tooth shape at the macro level, forms like hypsodonty, the ratio between the width and the height of teeth, that change at the evolutionary time scale. We can also change where you live. And obviously, in the evolutionary time scale, you can change your morphology to better suit where you live or move to a place where you're better suited to. And we can see changes in limb bones, horses' legs becoming longer, losing metapodials, both at the two-dimension and three-dimensional level. And again, we have changes in demographics, which is again in the ecological time scale, shifting of populations. So here's a good example from Boba and Beresmeheimer in 2004. And they're looking at, of course, the changes in number of Australopithecine, Homo, appearance of Homo and Paranthropines. And we look at the environmental shift, and we see that the grassland indicators, this classical, look, it's a GNU, a wildebeest, we have grasslands, kind of increases over time. There's a little dip here, but overall is greatly increased compared to the 4 million to, let's say, 2.8, right? And when we look at this distance, in this case, core distance, between the taxonomic data, which indicates a lot of fluctuations, which does not seem to mirror this grassland indicator, but our taphonomic indicators of what we see don't show again that much change. So if I had a take-home message, it would be like, let's start decoupling these things and not studying everything by themselves without, looking at the other factor, but realizing that change is not a complete box. Um, so just to show you a few, and I know I'm finishing up. So I think what I'm going to do is I'm going to leave all this my secret stuff that I just started to show you for next time we meet, and maybe it's a good place to change, and I can open the floor for questions so we can end on time.